Hi, my name is Alisa Yurovsky, and I'm going to present our work on sample-specific compartment deconvolution. Now, compartment deconvolution is a general algorithmic technique where you have a mixture and you have a bunch of features that have values, and the feature profiles are different across the different types that are present in this mixture. In our specific context, if you see on panel panel 1, I should say, we are interested in looking at patient biopsies, which are mixtures of different cell types. And for our context, for our motivation, please take a look at panel 2, where what we want to do is we want to be able to do differential expression analysis for the cancer studies. So for example, we want to look at specifically just the tumor tissue and see what differences we can find between two different groups of patients. Now, a simple, obvious answer to this is to use single cell sequencing to extract just the tumor cells for each of the patients, but that is still too expensive in the clinical setting. And so our idea is to build upon compartment deconvolution to get tissue-specific and sample-specific expressions. We can see this in panel 3. So you have two conditions, condition A and condition B. You have patients in each of the two groups, and you get their biopsies. Now, biopsies have different tumor purities, so they might have only a small or a large relative proportion of the actual tumor tissue, which is shown in blue for condition A and in more purple in condition B. So if we do bulk RNA sequencing on each of these biopsies, you're going to get these condition vectors, which give the gene expression for the bulk mixture. And so what you can see is it might be very often difficult to find significant differences in the tumor tissue by doing this bulk sequencing. Now, our idea, zooming into panel 4, is to extract for each of the patients the gene expression profile just for the tumor. So now for condition A, we have these blue cartoon profiles, and then for condition B, we have purple, and we can kind of see just by the difference in colors that we will be able to find the significant difference between these two conditions. So now for our approach, you can see in panel 5, we are using standard compartment deconvolution. Specifically, we're using a variant of non-negative matrix factorization. Our variant is called FASTA NMF, and it's going to appear in TCBB this year. So very briefly, you start with a mixture of gene expressions. So you have for each patient, for each of the genes, you have the value from bulk sequencing, and then you deconvolve it into a product of two matrices, W and H, where W is compartment or tissue-specific gene expression for each gene for each tissue, and then H is the proportion of each of these tissues in each sample. Next, we're doing our per sample gene optimization. So we take each sample, we take each gene separately, and we run an optimization where the objective is to minimize the error between the NMF-derived global W and this new W. Basically, we don't want to wander too far away from our general deconvolution result, but we want to get these patient-specific differences for each tissue. And then, of course, the constraint, that's a hard constraint, is that the new W values for each tissue will produce the original mixture values for that patient when multiplied by the H proportions. Panel 7 recapitulates our idea that with this two-step deconvolution, what we get is tissue-specific gene expression profile for each sample, and this will allow us to do interesting analyses. In panel 8, we see the results of our work compared to other methods on one data set, which is simulated data based, of course, on real experiments, which has ground truth, which is we know exactly the tissue-specific gene expressions for each sample here. And we see that our work shown in blue has lower error. We did another interesting small study recapitulating some results on the TCGA PAD data set. Basically, we took the data, we did the 3-tissue deconvolution using our method, and then we did consensus clustering on the stroma marker gene expressions from the stroma compartment, because we now have these values for each of the patient. And we were able to find survival differences for the stroma compartment for 2 main patient groups, which recapitulated the previous study. So next, the things that we will focus on is getting these compartment-specific marker genes where we don't actually know what the tissue is, so automating this process. Then we're going to apply this work to differential expression studies from some clinical data. And then we're going to continue refining this work because we feel that it has a great potential to advance personalized medicine. Thank you very much for your attention.